Okay. Hello all and welcome to the Computational Reproducibility Seminar series. Today, a Thursday at 11 o'clock, all new timing. I hope that not too many people were confused by this. I have the pleasure to give you today a distinguished speaker. From Swiss Transplant, we have Dr. Simon Schwab. I know Simon from my starting time at the Swiss Reproducibility Network when he was still doing a postdoc at University Zurich. And during this time, he was, well, can we say leading? I think at least pushing the working group on training. And one of the aspects when we speak about how can we train people to be more reproducible, the first thing we speak a lot about tools, especially also here in this computational reproducibility seminar. But the first thing they say is, can you give us sort of like a checklist on what to do and how to do it? How one of the students I had in my biomedical research course, when I gave the presentation on reproducibility, they said, yeah, but what can I now do short of learning how to program to become more reproducible? And as you might have read in the Slack, we're planning to have an event in autumn in Brieg for the working group. And we want to discuss about exactly these things, about checklists for computational reproducibility. And we're going to use the seminar to invite several speakers who already thought about these issues. What can be simple tricks and tips that we can use to become more reproducible. And today, Simon is going to speak about a more like high-level aspect of it, where reproducibility is one of the aspects towards it. And that is basically, he's going to speak about 10 simple rules for good research practice. Why can he do that? Well, because of his working experience as a statistician and having a PhD in health sciences from Uni Bern. I am super excited to have you here, Simon. Thanks for showing up. And the floor is all yours.
Thank you, Daniel. Very nice to be here in the Computational Reproducibility Seminar. And actually, I think this is the first time I present this paper. So first, let me just introduce where I work. So maybe you don't know it. Swiss Transplant is the Swiss National Foundation for Organ Donation and Transplantation, and manages the waiting list and is responsible for allocating organs in accordance with the law. And I work there as a statistician and have a lot of data related to transplantation. Now, transplantations happen in Switzerland at 6 centers. You can see them here. And you can also see which organs are transplanted at what center. And let me just mention the main sources of data that I'm using. So mainly we have the Swiss organ allocation system, where we have data on the transplant candidate and who received what organ from which donor. Then completely separate is the Swiss Transplant Court Study, which Swiss Transplant is not in charge. This is a separate organization led by, I think, University Hospital of Basel, and they collect all the data after transplantation. So all the organ recipients, they are followed up. So having said that, I want to acknowledge for this work I'm presenting here, the Foundation for scientific research from the University of Zurich who supported this project, and also the Center for Reproducible Science, which founded my position at that time, which is a competence center of the University of Zurich. So here's the paper. So actually, so I'm a first author on this paper, but actually I think this is the only paper where I'm a first author, which I didn't write. So this was a very collaborative effort from a diverse group of scientists from different fields like epidemiology, biostatistics, bioinformatics, psychology, biology, and so on, neuroscience as well. And we have together written that paper. And I think we assigned these 10 rules to the to different people. So I think I wrote one rule or two, but it was, I really want to emphasize this, it was a great collaborative effort. And I could never have written such a paper. I mean, I have not just the experience that is required, because the paper covers so many aspects, you know, from sample size, p-values, and study protocols, data management plans. So it's quite wide. Now, actually, I remember that on social media, somebody was not happy with the title of this paper because of the word rules. You know, rules is quite a strong word, and maybe it's not the right word. Maybe these are principles or guidelines or suggestions, you know. But here's the thing, when you write the 10 simple rules paper for post-computational biology, it has to have that word rule. So this was out of my power. And, you know, rules are rules. But of course, you know, I would rather see these rules as principles, maybe. So maybe a few things I want to emphasize. So how did this paper came up? Daniel already mentioned the course. So we created the Good Research Practice course at that time at the University of Zurich, together with Eva Forer and Leo Held. And so the idea was, why not write a paper? You know, because we were not sure, do we teach the right stuff? Are there things we forgot? You know, so why not ask a group of people from diverse fields what are the main topics we should cover, right? And the other thing I want to say is, what is the aim, or for which person this paper could be interesting? And it certainly is interesting for PhD students, early career researchers, you know, as a starting point. I mean, I would have loved to have such a paper as I was beginning my career. However, it can also be useful for others who want to do such a good research practice course or a related course on reproducibility. And also the examples is much flavored from clinical research, you know, maybe even biomedical research. However, I think a lot can be applied to other disciplines, like, for example, writing study protocols. This is as useful as in medicine, as well as in psychology or social sciences, and so on. And of course, this is not a fully exhaustive list. You know, there are other things as well which we did not write about. And what I also saw yesterday, as I looked at the paper again, we actually have more than 100 references. So it's a nice work to get into the topic and to look at other papers who then cover certain aspects in much more detail, you know, because here we could just be a little bit superficial, you know, and just touching these topics a little bit, you know. Okay, so when the first thing we did is we shortlisted all these rules, 10 rules. That's another rule. It had to be exactly 10. It couldn't be 8 or 12, you know. And so we had to find an agreement across all the authors. And once we had the agreement, you know, the first thing statisticians do when they see a list, they sort it, you know, usually alphabetically. Here we sorted them according to the research process, like planning, execution, and reporting. So you can see 5 rules for planning, 3 for execution, and 2 for reporting. Okay, the first rule, specify your research question. Well, sometimes as statisticians, you know, you ask, what is actually the research question? And then you get the answer from the clinician, the deadline is due tomorrow. So, I mean, what I want to, so this never happened to me, of course. But what I want to say by that is that sometimes research projects, the research question is not clear. And this is a little bit a pity. So it's an important aspect. The first thing in a project, you know, what's the question? And it's important to have a good research question. And I thought, well, how can you arrive at a relevant, good research question that matters? So maybe you just, you know, I thought maybe you just ask your PI, right, who is much more experienced. So talk to people who have more experience in the field, ask for their opinion. Is this good? Should we do that? Or there are also opinion papers, you know, like in the literature. And then some of the co-authors answered me, well, I think these are just opinions, you know. Maybe we should look at systematic assessment of research gaps. We should ask patients, you know, what do they think matters? And we should also involve the public. So I didn't know about these things. And I gave you here an example from my field, organ transplantation, led by Sonja Beckmann and Sabine de Geest. So this was a study where they asked patients and also professionals what do they think should be research priority, right? So they systematically assessed what are the research gaps, what do people think is important. So I find this wonderful. And in my work, in my own work, I have also involved patients. So I have 2 projects where in both of them, patients are involved. So organ recipients, and they are also a co-author on the publications. Another thing which is related to the research question is that sometimes researchers are a little bit confused about, do we do confirmatory research here or do we do exploratory? You know? So, and for me, it was just as confusing as well. So I thought maybe I could write a paper about that with Leo Held. And so basically in that paper, we write exploratory questions are as important as confirmatory research, but it's important not to mix it up, you know. So sometimes it can happen that researchers might present exploratory results as confirmatory, you know, to increase the probability of publication. So it's like a game of darts, you know, where the target is drawn around the dart only after it has been thrown. You know, it's a little bit like cheating, you know. And so there's also this joke from Steven Sen in one of his books. A medical statistician will not accept that Columbus discovered America because he said he was looking for India in the trial plan, right? So this was actually true. Columbus was looking for a route to India, you know, and then by accident, he discovered America. Of course, in clinical trials, everything has to be specified, you know, in a study protocol, and you cannot just change your plans, you know. But on the other hand, sometimes changing plans is needed, you know, not fundamentally, but you cannot foresee everything. You cannot plan everything, you know. So change plans should be a little bit flexible, you know. But I think it's more important to have a plan, to have a good study protocol, even though some things can change, and you will then also report these changes, of course, instead of having no plan at all, you know. And there's this other saying I like very much is when you fail to plan, I don't know who said that, when you fail to plan, you plan to fail. Okay, then the second rule is write and register a study protocol. So I do that in most of my projects, and here you can see a study protocol that we even published in a journal, the KIDMO study. So that's a the kidney prediction model, which I'm working on. And when I talk about registration, or sometimes it's also called pre-registration, sometimes people may object, you know, when I put my ideas out there and somebody may take it and do it themselves, you know, and publish it, and then I get scooped, you know. But actually, that's not true, because when you register a study protocol, it gets a timestamp. And so you can kind of prove I had the idea, I had the plan at that time. And the other thing is, you can put an embargo on a study protocol. So it's not openly available yet, but it's registered. And after the embargo, let's say after 1 year or 2 years, it will eventually get published, right? So that's the other thing. So this is not really a reason not to put things out there as a study protocol to get scooped, you know. Okay. Another thing I should mention are also registered reports. So I'm not very experienced with that, but I think it's a wonderful thing, because sometimes it's difficult, you know, to find a journal that publishes the results. And it gets rejected, and then you go to another journal. So it's a lot of waste, kind of. And with registered report, the journal says, well, we have accepted your protocol, and we will also publish your paper, no matter how the results will be. Then the third rule is justify your sample size. So I think there is a large body of evidence in the medical literature, but not only, that many studies are underpowered, so statistically underpowered. Or in other words, they don't have enough data, or not enough patients. And so sample size calculation is important. It's part of the planning. And there are great R packages for that. And it's really easy, you know, for example, sample size calculation for a t-test, the two sample t-test, it's very simple to do. Sometimes, you know, you submit to a journal, and then the reviewers say, well, did you do a sample size calculation? And then you do the sample size calculation after the study was done. So that's a big no-no, right? So it has to be done at the beginning, and should also be part of the study protocol. And sometimes in some settings, it's a little bit more difficult, you know, like, for example, in my area, prognostic models, prediction models, it's more difficult. It's not so straightforward like a t-test, but there's great help from researchers who created methodology and also our packages, like the group by Richard Riley from Birmingham, to calculate the sample size for prognostic models. Or actually, it's the other way around. So when you have a cohort study, you have your 2,000 patients, you know, that's fixed. You know the sample size. But the question is then, how large can my model be, or how many parameters I can estimate given that sample size, right? So it's really, really important in prediction models. And in some settings, it's quite, you know, I once had a statistical consultation from psychologists with such a complicated design. I hardly understood it. And there's no way how you can determine the sample size. So I had to do a little simulation. That's also really powerful. You can simulate the data under certain assumptions and then under other assumptions, which gives you a nice overview. And then you can look, how many times you find the result, the result, how many times do I find it? And if you find it in 80 times of 100 times, your power is 80%. So this is also really interesting to use simulations in more complex settings. Then the fourth rule is write a data management plan. And this is really the area where I have very little experience. I never wrote a data management plan. It's basically the data life cycle. So what data will be collected, how the data will be organized, stored, handled, and protected. You touch this in ethical approvals, but the data management plan is more detailed and a separate document, and is often required by funding organizations like the SNF, for example. But I understand that there are trainings for that topic, so this is really helpful. And another thing which is worth mentioning is the FAIR principle. So findable, accessible, interoperable, and reusable data. Then the fifth rule is reduce bias. So this is actually a topic where I know very little about. And then I started to give a talk about bias in medical research every year at the University Hospital of Zurich for cardiologists and epidemiologists. And every time I learned so much. And it's such an interesting topic, First, for me, it was more like, it's not about statistics, so it was more for me like an obscure topic, which I didn't know about. And now it's one of my favorite topics, actually. And there is this catalog of bias by Oxford University, where you can look up all these biases, how they are defined, and what can you do about it. So it's really a nice resource. There are so many biases, that's why I said every time I learn more. But here are the most common ones. And actually, the most important and common one we forgot. I think the most common one is selection bias. And I'll give you an example later. It's not here. This is when you make a selection of certain patients, and you do not include all of them. So that can lead to a bias, But there are many, many biases. Just one I find really interesting is immortal time bias, because it can pop up every here and then in the medical literature, and somebody says, "Oh, isn't here maybe immortal time bias?" It's sometimes difficult to detect, but basically it's when patients have to survive a certain time point, so to be enrolled in the study. So I'll give you an example. So it's like a textbook example. For example, COPD patients, they were assigned to the treatment group only if they fail a prescription in a pharmacy after discharge. So to get enrolled, you have to go to the pharmacy, right, after 3 months or so. And so in this time, you are immortal, because nobody, this is like a selection requirement to have survived that time. So it was impossible to die. And this introduces a bias, and the bias always works in the direction that the group with immortal time has a survival advantage. And so there was this study in Annals Internal Medicine about actors who won an Oscar have a longer survival, and it's immortal time bias, right? So this study is so the result is not valid, right? It's refuted. In my research, I also found biases, particularly in one of my projects about analyzing the wait list, and not just one bias, but multiple biases. So this was really interesting. And I just want to emphasize one of them. So when it comes to how long do you wait for an organ transplant, let's say for a heart, how do you calculate, how do you give an estimate, like for official statistics, an estimate of waiting time? So it's very easy, actually. So every organ recipient, let's say heart, one person waited half a year, one person waited 2 months, and another one waited 8 months. And you just take the average, and you have average waiting time, or you take, instead of the mean, you take the median, because this data is typically right skewed. But no matter what you do, it's wrong anyway, because you have selection bias. You have just patients that received an organ transplant. It's just the transplanted people. But you want to have an estimate that is useful for all the people on the waiting list, So that doesn't work because of selection bias. And the right way to do that is analyzing the data of all the people, like people who have not yet received a transplant, who are still waiting, and then are people who died on the wait list. You cannot ignore deaths. I mean, it's always a really bad idea to ignore deaths in medical statistics. And then you have also another group, individuals who were delisted, right? So you have to involve all of them to create an estimate of the waiting time. And the way you do that is to use methodology from survival analysis, computing risk, and you have a nice cumulative incidence curve, and then you look at 50%. So when the probability, at what time period the probability is 50%, and that's for heart. It's a bit less than a year, and for kidney, it's 3 years. So that's the waiting time estimated based on all the people and not just the transplanted ones. Then the next rule is a little bit controversial. Avoid questionable research practices. So this is a little bit, about all these bad things that you shouldn't do. The 10 Simple Rules paper is more on the positive note, like helpful, what should you do? And here, this is more about, the things like the sins. So we call that, I published that with Leo Held. So we call that the 7 things of significance test. And among them, p-hacking, this is a very common phrase, like torturing the data until you obtain the result that you like, these sorts of things that should be absolutely avoided. And to mitigate that, it's good to have a research plan. That's why study protocols are so important, where you predefine your plan. It's much more efficient. You could, in my project, sometimes I forget what to do. So I just take the study protocol, and then I can read what I have to do, what analysis I should do, and everything has been pre-specified. I really like that. So having said that, let's go to the next rule, and this is another controversial topic, which is p-values. I don't really like to talk about that, to be honest, because there is a lot of literature. I'm really not the expert on p-values. There are experts that published on that topic, whether it's good, it's bad, should be replaced, should we become all Bayesian statisticians? I cannot really say much about that, except I like p-values. I work with them. I try not putting too much emphasis on p-value. And the more and more they get less important for me. And I think in my last 2 papers, there are no p-values at all, I realized. But I don't, you know, I really like them. What I don't like is to threshold them at 0.05. You know, I don't like the phrase statistically significant so much. And then people might think I don't like p-values, but that's not true. I just don't like thresholding them, you know. And that's what I wrote here on LinkedIn, which got, to my surprise, quite a lot of likes and comments. I think there were like 2,000 likes and more than 100 comments, you know. And it is a topic that I think we always cover in these good research practice courses. It's important. And the main message is don't put, you know, don't worship the p-value. It's not so important, you know, especially not like whether it's significant or not, you know, like 0.0047 is not really different to 0.0051, but people may think when it's statistically significant, it's more important, but it's not, you know, it's important to report all the results and to report exact p-value. And this is what I wrote here in this post, basically, among some other stuff, you know. And what I also find interesting at maybe just one point of these many points here is that you can have 5 studies, each of them is statistically not significant, but together in a meta-analysis, you know, they can provide evidence for a treatment effect. So rule number 8 is make your research open. So open science, it involves open study protocols, which I already covered. Then open code. So all my R packages, I really like GitHub. You know, I work a lot with GitHub. I have 2 projects currently on GitHub where I work a lot on them. And then open data and open access. These 2 things I see a little bit different, you know, because in my field I'm working now, open data, you know, it's the data I work with is highly sensitive. So I cannot really, you know, put it on the internet. But researchers who are interested in it can request it, you know, for example, from the Swiss transplant cohort study, but it has to be a formal process. It's not like that this data can be then just put on, you know, put on OSF or all these repositories that you can put data to make it open, you know. So for me, the last 2 points are not so relevant for me. I really find protocols important and open code. Open data, it depends, you know, can you do it? I can't. And open access, I also see that really different now. You know, when you look at the processing charges, the article processing charges, you know, here you can see $12,000 for having a publication in Nature Reviews Neuroscience, I think, Nature Neuroscience. This, I mean, this is ridiculous, you know. So I try to find open access papers, but those with a reasonable price, you know. But what is a reasonable price? Certainly it's not 8,000 or more, you know. I find this is just totally overpriced, and you pay for the name Nature in it, you know. Absolutely. I really like the open science framework. So this is a wonderful tool to manage your research project. You know, you can register your study protocol. You can put your preprint up there. You can put data there, code. You know, it's really a great tool for researchers. And another thing where I focus a lot on is the analysis, making the analysis transparent and open. And therefore, I think the best possible tool to do to do that is notebooks, and my favorite is Quarto, and it unites code but also documentation. This is so important. I document a lot since I'm working with Quarto. What I'm doing, why I'm doing it, and also problems, like I have to ask this person because let's say we have a missing creatinine value for this patient. So I put it in there, and you can highlight it with callouts so it appears red, and then it's part, you have documented it and you don't forget it, because I work on different projects. So I tend to forget important aspects of the analysis. But with that, it's really reliable and well documented, transparent, and also reproducible. So when you have to rerun the analysis, you can basically press a button. And what I'm currently doing now is to all the official statistics from the national transplant activity. I want to, which involved a lot of manual work, I want to put it in Quarto so that it's all automatic and documented, and we have more time to do quality checks and improve the figures, and instead of doing like a lot of manual work, which might be boring. So that's my one single favorite tool with our studio and R. And I think it's the most, I think I recently read, I think while AI might be the most overrated tool, I believe Quarto is the most underrated tool at the moment. Then rule number 9, report all findings. So it is quite a scary problem that not all results are reported, especially when researchers find they are not so interesting to report because they're not statistically significant. And here you see a figure of set values, and you see this huge hole in the distribution, and which is the dark statistics. And when results are not published, like negative results in clinical research, this can lead to this can mislead decision-making and harm patients. And then the last rule, follow reporting guidelines. So the most known reporting guideline is, I think, CONSORT for randomized trials, because they are so regulated. There's so many rules, and what sometimes researchers might not know is that there are other reporting guidelines as well for observational studies, for prognostic studies, and so on. So there are much more. There's a great network called the Equator Network that collects and provides all these reporting guidelines, and you can download the checklist. So this is really something that improves research, and because you are more on the computational side, and myself, I work with prognostic models. Just recently, there is the new tripod guideline published. So that might be interesting for you because it now also involves AI machine learning, which was not covered so much. And this will replace, I understood, the tripod guideline entirely. So even though you do like a Cox model, which is not AI, you use this new tripod plus AI guideline. So that's the new guideline for all the clinical prediction models. So I think that's it.
Thank you, Simon. Fantastic talk. I enjoyed it a lot. There should be a clapping, yeah, like this clapping emoji.
Thank you.
As microphones usually cut out too loud noises when you actually do the clapping. So, yeah, very exciting. Unfortunately, I wanted to open with your LinkedIn statement. Uh, so now you already gave it, that AI is the most overrated and Quarto the most underrated tool. Sort of like, when I was starting my PhD, I was getting to learn LaTeX. A little bit later, I learned Sweave, S-Sweave. Cool people call it S-Sweave. And then our Markdown came around, and then our notebooks came around, and now we have Quarto. So that's what Phil Chang called the literate programming. So I try to teach my students that literate programming is a good practice to adhere to.